Yes, ma'am. So now we'll discuss about each and every unit in a very short span. So I will start from the lesson cell. Unit 3, I'll give some of the very important point. Kindly refer. You keep an NCERT also with you. Refer it. So let me start from the very basic of the discoverer of cell. So if we take discoverer, it is Robert Hooke, year 1665. We can say who discovered cell from cork of the plant or bark of the plant. Cork or bark, they are considered as a dead tissue. Later, Anton von Leeuwenhoek, in the year 1667, he discovered live cells. Remember examples like RBC, WBC, platelets, gametes, bacteria, and yeast. So, followed by discoverers, let us have a note on the cell theories. The very first cell theory given by Schleiden in the year 1838. Remember, years are very important. Who proposed a theory called plant cell theory. Whereas Schwann in the year 1839 proposed theory called animal cell theory, and he is the one plant cells has cell wall. It was discovered by Schwann. Though Schwann worked in animal cell theory, he is the first one to explain about cell wall of plant. Then Rudolf Virchow, 1855, who modified the first two theories and gave a complete cell theory. His theory says all organisms are made up of cell and new cells are coming from pre-existing cell. For this, there is a Latin term, omnis cellulae e cellulae. Question might come, the statement omnis cellulae e cellulae proposed by Rudolf Virchow, 1855. Then let us take the sizes of cell. RBC measured around 7 micrometer, bacteria 3 to 5 micrometer, mycoplasma the smallest known cell. Smallest known cell. Kindly read one or two line about mycoplasma. It is 0.3 micrometer. Longest cell in our body is nerve cell, whereas largest cell is egg of an ostrich. Don't consider virus here. Virus is not a living organism. Virus is not classified under the five kingdom method. The five kingdoms are Monera, Protista, fungi, Plantia, and Animalia. Viruses are not kept anywhere. If we take bacteria, based on the shape, we can classify bacteria. There are different kind of shapes are there, as rod shape, spherical shape, comma shape, and spiral. Recall the example I have mentioned. Rod shape is Bacillus, spherical is Coccus, comma shape is Vibrio, and spiral shape is Spirillum. If we take a prokaryotic cell, example of prokaryote is bacteria. All bacteria are prokaryote. If we take example of an eukaryote, it's very simple. Anything other than bacteria, all are called as a eukaryote. Organelles or structure found in prokaryotes. Nothing but cell envelope. This is the very first structure. Cell envelope itself is made up of three layers. Outermost layer called glycocalyx, middle layer called cell wall, and innermost layer called as plasma membrane. The structure of plasma membrane of a prokaryote is equivalent to the structure of plasma membrane of an eukaryote. This is a very important point. Many times, repeatedly question came from this. If we take the glycocalyx, it differs in different kind of bacteria. It might be loosely arranged, we call it as slime, or it might be tightly arranged, we call it as a capsule. The second organelle is mesosome and chromatophore. These are the inward folding of plasma membrane, and mesosome is very important for bacteria, where all the major metabolic activity like energy production, nothing but respiration, cell wall formation, DNA replication, sharing of DNA to daughter cell, all the metabolic activity are done through mesosome. And chromatophore is also inward folding of only photosynthetic bacteria. Example for photosynthetic is cyanobacteria. Cyanobacteria, commonly called as blue-green algae. We have read in lesson microbes in human welfare, in biofertilizer, the last example, cyanobacteria. In the case of a paddy field, they increases soil nitrogen and they add organic content to soil, which increases the fertility of soil. So chromatophore found only in photosynthetic organisms. Then the third organelle is nucleoid. Nucleoid is equivalent to nucleus of an eukaryote, but it is a non-membrane bound. That is why we call it as a nucleoid. In the cytoplasm, are also called a cytosol. Remember, cytoplasm is the major arena or site for biochemical activities. The genetic material of a bacteria will be in two sets. One is called as a chromosomal DNA, also called genomic DNA. The other one is called plasmid DNA. Chromosomal DNA is the actual one. Plasmid is an extra copy. Remember, definition is very, very important. Plasmid can be defined as extra copy of circular DNA. Extra copy of chromosomal DNA, which is circular in nature. They are providing a special phenotypic character to the bacteria, which is none other than antibiotic resistance. Remember the example PBR 322 from the lesson Biotechnology Principles and Process.